Okay, Lisa Chadwick, Program Director in Genomic Sciences, is now going to take you through a concept for Mendelian Genomics. Go ahead, Lisa.
Okay, so yeah, like Rudy said, this is a concept for two RFAs that represent NHGRI's continuing investment in Mendelian gene discovery. So before I start, I just want to take you back through the history of Mendelian gene discovery over the years. And I took these data from these two papers referenced below, one of which just came out like last week. One of the figures in those papers compared the rate of gene discovery with conventional methods versus more advanced methods. So as you can see, back in the day when we used to do linkage mapping and positional cloning, it did take a while to identify the genes that underlie Mendelian diseases. But you can see it's sort of, is this a pointer? Does that even matter? No. Okay, sorry. I'm just not even going to point. You can see that things sort of leveled off to where we were identifying the gene underlying, about 170 on average Mendelian diseases or phenotypes every year. And then in around 2010, people started to apply things like whole exome sequencing to Mendelian gene discovery, which really turned this into more of a computational problem. And you can also see that significantly bumped up the discovery rate. And so now we're able to identify the genes underlying these Mendelian phenotypes, about almost 300 of them every year. So as you have all heard, oh my gosh, I keep going forward. As you've all heard in these strategic planning discussions, we've been asking ourselves at NHGRI, what represents the forefront of genomics now? And we're asking those same questions in the context of our Mendelian Genomics program. Now that we really understand how to find Mendelian genes using whole exome sequencing, what is the forefront for Mendelian genomics? And so first I would say that novel gene discovery is still a really important part of that forefront. So these are data from the Centers for Mendelian Genomics program, which has been running for 8 years. And what I want you to take away from this is that the rate of discovery has really not slowed down. So we don't appear to have reached sort of the plateau of Mendelian gene discovery. And this falls in line with what you see here. This is data that our coordinating center has taken from OMIM. But of the roughly 8,000 currently delineated Mendelian phenotypes, we only know the genetic basis of about 65% of them. And that represents less than 20% of the genes in the human genome. And of that, a quarter of those actually underlie more than one Mendelian phenotype. And you would expect that a lot more of those genes, when disrupted, would cause some sort of disease. And so taken together, this all suggests that there's still a lot more to do in terms of novel discovery in this area. But I also want to talk to you about this problem, which I think is a big part of the forefront for Mendelian genomics. And that is figuring out how to solve those cases that we can't solve using whole exome sequencing. So these data, again, come from the Centers for Mendelian Genomics program. They're pretty representative of other rare disease efforts or what you see in clinical diagnostic labs. So these are the phenotypes that we've looked at in the CMG program where we were able to make a tier 1 discovery where we would call that really solved. We have multiple families that have the same variant underlying the phenotype, or we have a linkage peak and some additional evidence that really makes us feel convinced that that is the variant that underlies the phenotype. So that's about half of the phenotypes that we've looked at in the program. And then we have this part of the pie chart. These are the tier 2 status variants. So this is a suggestive association. It looks like it could be the variant that underlies the phenotype, but we probably only have one family that has this, and we don't have that additional sort of functional follow-up to back up that finding. And then we have this other quarter of the pie chart, which are those phenotypes where we did whole exome sequencing and we were not able to identify the underlying genetic cause of those. And so together, those tier 2 and this category represents almost half of the phenotypes that we look at in Mendelian programs. And so what I would argue is that figuring out how to solve those kinds of cases that you can't solve easily using whole exome sequencing is something that's really at the forefront of Mendelian genomics. And so how do we move from a situation where we can only identify half of them to where we're able to fill in most of this pie chart? So what are the things that are needed to get us to that point? Well, first, I want to say that data sharing is important for all aspects of this. So moving both of those categories over to that tier 1 solved state. You need multiple families or multiple individuals with that phenotype and that variant. And the one problem with rare disease is that it's rare, and those cases are spread all over the world. They may have been sequenced, but their data is in some clinical labs database or in some medical laboratory or in a program like the CMGs, and it's hard for those patients to get, for those data to get together so that we can say, oh, we actually have more than one family that has this variant associated with this phenotype. There are some ways to do that, things like matchmaker exchange, but even that has a lot of limitations. And so I think that data sharing is really important for helping move us towards that place where we're able to solve most of the cases that we look at. Another thing that's really important for this is functional follow-up. So even if we have only one family that has a variant associated with a phenotype, we are able to move that to that tier 1 or solved status if we're able to do functional follow-up that convinces us that that variant actually impacts protein function in a way that makes biological sense, or that it leads to a similar phenotype in a model organism. So this is another aspect of being able to move something into a place where we can call it really solved. And then we also know that whole exome sequencing, by definition, is only looking at a portion of the genome. And there are a lot of other important parts of the genome that may be involved in Mendelian phenotypes. So we may need to apply more sequencing or different sequencing, like whole genome or RNA-seq or looking at DNA methylation to help us understand the genetic cause of those phenotypes. Now, when you have all that additional sequence data, you also have a lot more variants to sort through, and it's hard to prioritize which ones of these might actually be important. So we need to develop high throughput ways to help us prioritize those non-coding variants so that people know which ones to follow up on. So what we're proposing here is a Mendelian Genomics program that's going to help us get to this place where we're able to solve most of the phenotypes that we look at. Because novel discovery is so important, we will continue to have a focus on novel discovery, but we want to place a greater emphasis on solving those cases that aren't easily solved by whole exome sequencing. We want to allow the groups to do that functional follow-up on candidate variants that will help them move things towards that tier 1 solved state. Because data sharing is so important, we're going to place a really high priority on data sharing in this program. We also want the groups to work together, and we want to bring in the broader community through the use of opportunity funds, which I'll talk about in a moment. And so we're going to accomplish this with 2 RFAs, one for Mendelian Genomics centers and one for a data coordination center. So the Mendelian Genomics centers will be where they'll be using sequencing to identify those novel gene phenotype associations underlying those Mendelian conditions. We want them to work to develop scalable approaches to do those important functional follow-up experiments. To help us achieve that goal of broad data sharing, we want them to place a priority on samples that have detailed phenotype information that they're able to share and that have been consented for broad research use. It's important to work together, so we want them to work together as a consortium to help move cases towards that tier 1 solved status. So we envision that we would have working groups within the consortium where they'd be working together on things like unsolved cases or ways to do variant interpretation or best practices for those functional validation experiments. Taking these findings to the broader research community is also really important, so we would like these centers to engage and empower that community through outreach and education efforts. And then we also propose a data coordination center for this program. That's going to help us achieve that goal of data sharing by ensuring that quality-controlled, complete data from the genomic centers is made available to the community through things like the Anvil, ClinVar, Matchmaker Exchange, et cetera. because we want the centers to all know what samples are in each other's pipelines so that we can pool them together when needed. So the Data Coordination Center will help track those collaborations and samples in the pipeline. We also want them to think about a way to create some infrastructure specifically to share sequence and phenotype data from those unsolved cases, not just within the consortium, but with the broader community, and ideally in such a way that other programs or other efforts could put their data in that same sort of framework. The Data Coordination Center will also be responsible for the logistical coordination that is so important for a consortium like this to function. And then they will oversee the Mendelian Genomics Opportunity Fund, which would sound really novel if Mike hadn't just talked to you about opportunity funds. But this provides some flexible funding for outside investigators to do things like validation studies, work on annotation or new analysis methods, or other opportunities as they come up. And so again, these kinds of funds allow for funding of small projects with a more rapid timeline than traditional grant mechanisms. They give us that flexibility to address current and emerging areas of need within the program. They'd be administered as subawards through the DCC. The Data Coordination Center coordinates the intake of applications and their review. They would be peer-reviewed in some sort of NIH-style review panel. And there, as Mike mentioned, there's a lot of programs that have used opportunity funds, including the UDN and the 40-nucleome program, which I was involved in before this. And then I wanted to close with the proposed funding levels for this program. So over 5 years, this would be a $60 million investment of roughly $12 million per year. We envision that $9 million of that will go to fund 3 to 4 Mendelian Genomics centers. And then the remainder would be used for the Data Coordination Center and those opportunity funds, which we envision would be roughly $750,000 per year. And so those are the slides that I have, and now I'm happy to take your questions or have discussion about this.
So I think it's hard to argue that the CMG program has not been tremendously successful. Back when that was being conceived, there was an emphasis on sort of a brute force approach to Mendelian disorders. So you aim to recruit centers with overlapping, substantially redundant expertise. Let's bring in lots of samples, let's sequence them, let's analyze them. Now you're asking, I think, something very different. You're saying, okay, we now need to step back. There's things that have been unsolvable, and how can we move them to the solved category? And I think that that's going to take complementary expertise, not necessarily redundant expertise. You know, the factors contributing to the unsolvable include things like non-coding variation, oligogenic inheritance, multifactorial penetrance, superficial or nonspecific phenotypes. So you can imagine now wanting one center that's going to do targeted recruitment of patients with a particularly irksome phenotype, do deep granular phenotyping to segregate those patients into subgroups that would now be amenable to giving answers, while another center is going to be really good at analyzing genomic non-coding variation. So I guess my biggest reservation is about how to do the application process for this. Do you ask for a bunch of centers blind to what anybody else is proposing to just throw in something, and then NHGRI will create a Frankenstein consortium at the end of that? Or do you ask centers to contact each other to identify areas of complementary or synergistic expertise and to propose something that's grander than the sum of its parts?
Well, so we'll lay out in the RFA what kinds of things we're looking for from these centers. They will have some flexibility to have a specific area of focus, just like the things that you mentioned. this is a problem that I think faces all consortia when they come together. We basically tell you, come in with your best ideas, and then we're all going to sit down afterwards and we're going to figure out how these work together. Usually it works pretty well. I think we do want to get a diversity of approaches and a diversity of ideas. And so that's the best way to do that, I think, is by allowing for that open competition with people being able to bring in whatever they think is the best approach.
Let's say that you get 4 applications that just say we're going to sequence more to solve this, and none that say we're going to focus on the pitfalls of phenotyping.
Right. So
How do you?
first, I hope that those all wouldn't do as well in review because we're specifically asking for things that are bringing in new approaches. So some part of that comes down to the review and programmatic decision making. But in general, those are things that we'll write into the RFA and to the review criteria that will hopefully help avoid a situation like that.
So I'm very, very supportive of it as a member of the advisory panel for the genome sequencing program. I think we have an official name somewhere. Adam probably knows what it is. This is the CMGs have been viewed as highly successful, perhaps even wildly successful. And as you pointed out, there's not been a plateauing in the novel discovery. And while I am very, very supportive of trying to figure out how to get to tier 2 to tier 1 and the unknowns somewhere, into tier 2 or tier 1, you don't want to take away from the novel discovery aspect because it's still a lot there to be done. And I realize you're highlighting that, but I was a little worried when I read the concept clearance that the solving the unsolved was getting a higher priority than continuing to find the novel ones. And I just wanted to make the case that we really should be doing both. I agree,
Yeah.
but the novel discovery should be at least as important as the other. And following on that, it seems to me that there's quite a bit of money going into the coordinating center, and the actual money going into the Mendelian centers seems a little bit less than where it is now, and that seems to me to be perhaps a little bit backwards. I would be saying you should be putting at least as much money as you are now into the actual centers and adding the coordinating center to it.
Right.
So I would be advocating for a higher budget, actually.
Well, first of all, I'm not going to argue with you about a higher budget. If you guys would like to give us some more money from somewhere, that'd be great. But no, I understand what you're saying. And we thought about that really long and hard. I think I could not in good conscience stand up here and talk to you about how important data sharing is for Mendelian gene discovery without making a real effort to make sure that our data get shared. And I think a data coordination center is going to be really instrumental in making sure that that pipeline continues and things get out. You know that that's an issue that we've struggled with in the CMG program. We're working really hard to get our data into Anvil, but it's a process. And so we really think that having a data coordination center like this is going to be important for helping achieve that goal of data sharing.
I absolutely agree about the data sharing, and I think it's really important going forward to try to be able to generate the sample sets that are easily and more easily shared with a better phenotyping on a lot of those than what has been there. I totally agree with that, but that's just another thing that needs to be done in this program because it's all really, really high priorities and has all been very, very successful, and I really would like to see it continue.
I guess two different comments. One is we've talked a lot today about coordinating centers, and I really do think perhaps some systematic review of all the coordinating centers, and they all seem to be getting set up slightly differently with slightly different RFAs,
Right.
and given the amount of funds going into them, maybe really looking as kind of a nitpicky part of the strategic plan of how coordinating centers get structured and funded, like data sharing should be done the same way across multiple. I just worry that in my own experience with my part of NHGRI, they're all slightly different. And I do think it's important to really put some effort on that behind the scenes. I do think it will be interesting to see how to judge the discovery versus the hard cases. So, I mean, I had the pleasure in the way of being co-author on a paper that solved a known syndrome that we started studying 20 years ago. And it turned out it was a weird allele deep in an intron that somebody really smart, taking data we had sitting around for a while, kind of finally figured out. But those are really hard to measure.
Right.
Compared to I'm just going to sequence more families. And so I think it will be really helpful to think for the review panel, how are you, some way of having all the reviewers kind of weigh those things the same.
Yep.
At least some benchmarking or something, because it's a lot easier to say, I'm just going to do more families and do exomes and get lots of new discoveries, and I'm not going to focus. So I know you try in the writing to balance them, but it'll be really helpful for the reviewers, I think, to think about how that's going to be done.
Yeah, no, those are really helpful comments. And I also understand what you're saying about the proliferation of data coordinating centers. I think one thing that is helpful is that now we have the Anvil. So we are not envisioning for this coordinating center for sure to be a separate place, right? So they're actually just going to help the data get to Anvil. So that's hopefully going to help centralize things across multiple programs.
So this is just to emphasize some of the prior comments. So having submitted samples and knowing about the CMGs, a lot of those cases I'm not convinced are simple Mendelian cases, and that's why they're not solved yet at this point.
Right.
And to be able to understand those, I think it's a matter of seeing a lot more data. And so I think it'll be on Anvil, but I want to emphasize the point. I think it's putting together the CMGs, the UDNs, the Gabriella Miller Kids First, as many of these different groups into one place, because I do think there are overlapping groups within those and you need the numbers.
Yep.
So I hope that's part of what's planned going forward.
Yeah.
Could you give just a little bit more detail about the opportunity funds, the review, the peer review, how that's going to be set up, and who do you think is actually going to be doing those peer reviews?
Right. So I can tell you about my experience in the 4D nucleome program, and I envision that this would go a very similar direction. So what happened there was the organizational hub got the applications in. They worked to find a peer review panel of people outside of the consortium, just in the same way that CSR would do. We did an editorial style review to start with, and then we had a discussion after that. And then basically we got scores just the same way we would have from the NIH review. It just takes less time, and it gives you more flexibility to say, okay, this is a current area of need in the program, and we can get applications in that will address that need and fund it more quickly.
So just reflecting on some of the comments that Jonathan made, much of the new discovery is in the category of N of 3 early onset, profoundly severe de novo conditions. So there is a lot more of that to do. But in the unsolved category, there's common Mendelian phenotypes with reduced penetrance and high locus heterogeneity. We know nothing, essentially, about bicuspid aortic valve. It's just not giving up its secrets. And to some extent, I think tackling bicuspid valve is more important than understanding every N of 3 phenotype. Ideally, I'd like to do them both, but I don't want to de-emphasize solving those bugaboos that really are going to make a difference from a public health perspective.
I totally agree with that, but I think both are really, really important. I think you don't know what that N of 3 might tell you about some basic biology that's going on. So I don't want to de-emphasize that. I think we need to emphasize what you're talking about as well, and which is why I'm saying, maybe there needs to be more emphasis on this program.
But just to follow up on Hal's point specifically, so how will Anvil, for instance, work with data stage? Because just specifically the bicuspid valves, most of those data, genomic data, are going to be in NHLBI. And to be able to solve those cases, you're going to need to combine the data from the two groups or from multiple groups.
Luckily, I see Valentina standing up because I don't know the answer to that.
So we are establishing interoperability processes with data stage. We're at the very early stages, as you can imagine, and Kids First is going to be part of the same conversations. And in parallel, NIH, with the nomination of Susan Gregorick as the Associate Director of Data Science, will also get back to facilitating this type of interactions. We're not waiting for it. We're having our own ongoing conversations. We have a meeting in early October of Data Stage, Kids First, and some other projects in the Common Fund group, as well as with NCI. So we're starting to have these conversations. So it's coming, but we're going to do it through the Anvil, not through, one data coordinating center and another data coordinating center in different programs.
So one last thing, the word scalable when partnered with functional analyses makes me cringe a little bit. So I would want to avoid the suggestion that it's a one shoe fits all kind of thing, and more of strategies that are tailored to be informative for that specific indication.
Right.
The Care for Rare consortium up in Canada has done an interesting thing of sort of partnering gene discovery labs with the lab doing the best basic science or best model organism work on
Right.
that gene or the
So that's one of the main reasons for those opportunity funds, because they allow us to say, these are some high priority things that we want follow up on, and we need the experts to do that. Come on in with your application. So that's exactly why we did that. I think that there are some things that you can do on a more high throughput kind of scale that are a little bit more generalizable, but you're absolutely right. There are definitely things that we're going to need to bring in more expertise on, and that's what we plan to use those for.
Okay. Comfortable with one vote, or do you want two? One vote? Good. All right. Can I get a motion to approve the concept for these two RFAs? A second. All in favor? Any opposed? Anyone abstaining? Thank you. Thank you, Lisa.